Well, welcome everybody. My name is Beth Foss, and I am the Director of Operations at the Choroideremia Research Foundation. So excited to have you. Welcome to 2023. Can you believe it? We're going to be doing a new series that will be commencing now and for the next few months on new therapeutics for potential treatments to choroideremia. And it's really exciting. So today we have a great guest with us, Divya Sinha. Dr. Divya Sinha works with Dr. David Gamm, University of Wisconsin Madison, and she also sits on our science advisory board. So we're very excited to have her here. And we're also really encouraged to have all of you here, whether you're watching it live. Wow, 30 people right now still coming in. Or whether you watch it on our YouTube channel. We just hope that you find these new therapeutics both educational and encouraging for the next year. Lots of things going on, lots of hope. So we wanted to say thank you. Thanks for coming. And a couple of the ground rules, just as a reminder, we want to first of all thank our sponsor, 4DMT, for their funding to help promote these videos. We are going to be using the chat feature like always as Dr. Divya presents her material and her PowerPoint. I'll be watching the comments and I'll ask the questions on your behalf. And I think that's it. Make sure. Yeah, I think it is. So I have to ask you, being a Minnesota girl, knowing Minnesota in January is extremely cold. How's it up in Wisconsin and Madison?
It was actually a bit warm, but we had some freezing rain and then snow last night. So it's kind of white again outside.
Oh, that's awesome.
Yeah.
Stay warm for sure.
Yeah.
Well, I'll go ahead and turn off my video and we'll go ahead and let you start and get started. Thank you.
Sure. I'll start sharing my screen. And I hope everyone can see this slideshow.
You're good.
Okay, awesome. Hello, everyone. It is my pleasure to be here today. And before I get started, I want to thank the Choroideremia Research Foundation for this wonderful opportunity to talk about two different types of potential therapeutics for choroideremia. So there are multiple avenues being explored. I'll be talking about two different approaches today. And as Beth mentioned, I'm a scientist in Dr. David Gamm's lab at the University of Wisconsin Madison. What we do is we utilize human pluripotent stem cells, and utilizing these cells, we derive cells of the retina in a dish, and we apply these cells for disease modeling for various retinal degenerative diseases, including choroideremia. And we also use these cells for testing different kinds of therapeutics. So two of the therapeutics that we work on in the lab are DNA editing and read-through therapies. So that's what I'll be talking about today. And before I dive deep into these therapeutics, I wanted to first give like a brief overview, the basics of what's happening at a molecular level within a cell when it's reading a genetic code and what goes awry in choroideremia. So in the eye, there's this thin tissue at the back of the eye. It's called the retina. And retina is comprised of multiple different cell types. We are going to focus on two different cell types. One is a neuronal cell type called the photoreceptors that receives the light, converts into electrical signal, and so it's transmitted to the brain, and so we have a visual. There's another cell type adjacent to it. It's called the retinal pigment epithelium, or RPE cells. And these cells play a variety of roles, and they help with the maintenance of these photoreceptors. So you can imagine anything going wrong with the RPE cells or the photoreceptors would affect vision. And what happens in choroideremia is that the RPE and photoreceptor cells are affected. Now, more and more, it's thought that the primary effect is in the RPE. There's still more work being done about that. But overall, what happens is that the RPE cells are dysfunctional and the photoreceptors also start degenerating. So this is just a schematic for what kind of happens at early stage of disease. So some of these photoreceptor cells start dying, and individuals start gradually losing vision. And because there are cells still present here, what can be done as a therapeutic approach would be to provide some kind of means so that the cellular function can be restored. So the cells are already there. We provide a therapeutic to the cell, and we would expect the cells to start functioning properly. That's for early stage of the disease. However, what happens during late stages is that a lot of cells have degenerated. So there are no cells available where therapeutics can be, which can uptake the therapeutics, and it can allow restoration of cellular function. So in such cases, for late stages, the approach would be to utilize cell replacement therapy. So that's like some labs are working on that, including our lab. And so this is not what I'm going to talk about today, but I did want to mention that the therapeutics I'm going to talk about today are expected to work early to mid stage of the disease when the cells are still present in the retina. And so to understand what's happening in choroideremia, let's look at like a cell. So if we zoom into a cell, say a photoreceptor or an RPE cell, each cell has a nucleus, and the DNA is packaged in the form of a chromosome. So if we keep zooming in, what happens is that if we kind of unwind the DNA, we can see it's formed of multiple genes. It consists of multiple genes. And one of those genes is the CHM gene. This gene encodes for a protein called Rab escort protein 1 or REP1. However, there are some changes that can happen in the genetic code, which are called the mutations. And because of these mutations, the genetic product is not available for normal functioning. So the REP1 protein does not function properly. So understanding how this genetic code works, and I wanted to go over this again so you know like where the therapeutics are working and what kind of outcome do we expect from that. So for example, we all speak English, and we have 26 different alphabets. Say I told you that you have to make a sentence using 3 letters at a time. So some examples for these 3 letter words would be, say, if I said R-A-T, rat, you would immediately know what it means. Similarly, if I told you C-A-T, cat, you would immediately know what it's referring to, what animal it's referring to. It's referring to a cat. And if I change the order a little bit, the meaning also changes. So if I said, instead of R-A-T, I said A-R-T, you immediately think of an art piece, right? And so similarly, what happens in a cell is that there are 4 different types of molecules called the nucleotides. And you might have seen these letters, AGCU or AGCT. U is used for RNA molecule and T is used for DNA. So for this talk, mostly I would be, so wherever you see DNA, you'll be seeing T instead of U, and then for RNA, you'll be seeing U. AGC and are common. So these nucleotides, we have coded them with these letters. And what happens is that, just like I gave you example of 3-letter words, the cell reads these nucleotides in sets of 3. These are called a codon. Using these 4 different nucleotides, if we end up making 3-letter sets, there are 64 codons possible. And so here's a table. You don't need to remember that, but it's just to show you that different codons refer to different units that make the protein. So these units are called amino acids. And so out of 64, 61 of them make amino acids, but 3 of them are called something called stop codon because they do not code for any amino acid. What happens then is that, say, if you look at this gene over here, so there's a start to the gene, there's a stop sequence in the gene, and the stop sequence is the stop codon, the sequence that encodes the stop codon. And so in the cell, what happens is that for a gene, for example, CHM gene, there's a two-step process to make the protein. First step is called transcription. And so another type of nucleic acid is made. It's called the RNA. This is called the messenger RNA because it's carrying the message, but it's not the final product. What happens then is that once this messenger RNA is formed, the cells then proceed to follow up with another process, which is called translation. So the message in the messenger RNA is now getting translated to get the protein product. And to decipher this message, there's other types of special molecules called the transfer RNAs or tRNAs. You might have heard of this term before. And so I'm just showing a couple examples of possible, cartoons for what a tRNA might be. So what a tRNA does is that it has opposite sequence. So it's like you can imagine like fitting in puzzle pieces. And so it has a set of 3 nucleotides, which are called anticodons. And these anticodons can recognize the codons and bind there. When it binds there, the transfer RNAs or the tRNAs also bring amino acids with them, which are the building blocks of the protein that the cell is making using this message. And so the tRNA will identify the codon, bring the amino acid with it, and then the next tRNA comes in with another amino acid based on the codon this tRNA identifies. The units keep attaching to each other, and each new tRNA that comes in keeps on carrying the amino acid from the previous tRNAs that have been joined now to form like a chain. And so this process keeps going. The protein or the polypeptide chain keeps growing, and eventually it's going to hit this stop over here, for which there's no tRNA. So the cell says, okay, so the message has ended, we need to stop. And so then the protein is released and the fully functional protein is formed. What happens then is that in some cases, if there are mutations, for example, if there are mutations in the CHM gene, they affect the formation of this full-length functional protein. So a full-length protein is not formed, and when it's not there, it cannot perform its function. And so I'm going to give a couple of examples of these mutations. There are multiple different types of mutations, but just to give you an idea, like how the change in these codes can affect the formation of protein, I'm using 2 examples. One is the effect of missense mutation. So as the name suggests, whatever the codon was supposed to relay the message, it changes. And so, for example, if instead of saying cat, like CAT, if I change the T to an R, now you have a car. So the message completely changed. You would not know I was talking about a cat because this is what you are envisioning when I say, when I change the message. This is what happens in case of missense mutations. So what happens in the gene, there is a change in one of the nucleotides. And as the tRNAs come and they are reading this message at a particular location has changed. So it's going to put in a different amino acid instead of the original one. And that's why then in that case, the protein function changes, or the protein is not functional anymore, because the residue that's required for it to form the proper structure is not there. And so that's an example of missense mutation. Similarly, let's look at another mutation type called the nonsense mutations. Again, as the name suggests, it does not make sense. And so giving you another example, if I told you rat, you immediately think of this rodent. However, if I change it to RRT, you'd be like, what does that mean? That doesn't mean anything, right? And so similarly, when there's a mutation, it leads the codon to change to a stop codon. When the transcription happens, the RNA is made, the messenger RNA. When translation is happening, there's no tRNA binding to this new stop codon. And so the protein formation stops early on, and the truncated protein is formed. Because this is not a full-length protein, it's not functional either. And so these are two examples of mutations that happen in choroideremia. And about 1/3 of individuals affected by choroideremia have this particular type of, like, they have nonsense mutations. And so moving over to different kinds of therapeutics that can be applied to choroideremia, I'll be talking about DNA editing today and translational read-through. So what DNA editing does is it involves correction of the genetic code, although you can use it for silencing as well, sometimes silencing of the gene. And then the translational read-through is another approach which involves modification of the cellular reading machinery, so it can read the nonsense mutations as well. So here's an example. I just showed you the DNA is first transcribed to RNA, which is like a messenger, and then this RNA is translated to protein. So when I talk about DNA editing, we are targeting the DNA and making a change at the genetic level. When I talk about translational read-through, I'm talking about using the RNA and making a change or making a change at the RNA level. So we are not doing any kind of genetic changes here. And in both cases, we end up getting the expectation is to get functional protein. So let's start with DNA editing first. These are some of the questions that I'll be going over. And I've tried to keep it simple. There are like some nuances, there are more details that if anyone has questions about, I'm happy to go over. But I've tried to give like a basic overview of what these technologies mean. So I'll go over what is DNA editing, how is DNA editing used to target specific mutation, and then I'll talk about, can all mutations be corrected using this approach? Finally, what about off-target cutting and editing in the human genome? Because we only want to target the mutation. We do not want to start cutting and editing at loci that we don't want to get affected.
Dr. Sinha, can I just ask a quick question? We had two that just came in, and if I
Sure.
could, before you get started. So Lisa, hey Lisa, she said, hi, I'm just wondering, can these therapeutics be applied or used to all mutations, or is there one therapeutic method for each specific different mutation? And thank you.
Yeah, and that's a great question. So DNA editing can theoretically apply to all different kinds of mutations because you're changing the genetic code. There are some technical difficulties. I'll talk about the considerations, but there are some technical difficulties because you have to have this particular molecule to target the locus that you want to change. And sometimes just because of how the bases are, it's not possible to do that. But again, theoretically, it's possible to target all types of mutations using DNA editing. However, the read-through would only apply to nonsense mutations. So when instead of like a normal codon, there's a stop codon because of the nucleotide change. That's when read-through therapy can be applied. Read-through cannot be applied to any mutation that's happening at a splice site or a missense mutation and so on.
You, I think, in turn answered this, but I'm going to go ahead. There's another question. Would DNA editing work for those with CHM who have a deletion?
It should work for them as well. It becomes a little more complicated, and I'll talk about that because there's, for those kind of corrections, there's a special template that's required. And the more types of molecules we add to the therapeutic, more complicated it becomes to like deliver them. So again, theoretically, it is possible to correct the deletion if we can provide a template for correction, but it can be a little challenging.
Thank you.
Yeah, sure. So going into DNA editing, first let's understand what is it. So again, as the name suggests, we are doing edits in the DNA, into the genetic material of the cell. It can be used for correction of a mutation, or it can be used for silencing of a mutant gene. The silencing portion is a little bit, so it goes with like how the repair machinery is working. So I'm not going to talk much about this, but if anyone is interested, I could go over this. But this is not expected to apply to choroideremia because there's mostly males are affected and there's just a single gene, unless the gene is to be silenced and replaced with a functioning gene. But mostly we are looking for correction of mutation. What happens with editing is we are looking to insert some nucleotides or replace the incorrect ones with the correct ones, or remove portions of DNA sections. So those are the things that can be done with DNA editing. It can be accomplished via different methods of like what approach we're using for altering the DNA sequence. So I'm not going to go into details of each one of them, but I did want to mention that in case you hear like, okay, somebody's talking about gene editing with CRISPR-Cas9, someone's talking about base editing, someone's talking about prime editing. What are these different things? It's essentially all under the umbrella of DNA editing. It's just like what you're editing and what your approach is to edit. These names are based off of that. So gene editing using CRISPR-Cas9 can be used for correction or silencing. Base editing is typically used for correction because the base refers to a single nucleotide. And so you can change a single base back to what it's supposed to be. And then prime editing is more newer approach, which allows like more precise editing without a lot of nearby off targets. So base editing and prime editing are mainly used for correcting the genetic material, the genetic mutation. And so how do we target a mutation site? So as you already heard, I talked about messenger RNA. I talked about transfer RNA. So RNA plays a pretty important role. And there's another molecule. It's again, a nucleic acid. It's a small nucleic acid. It's called the guide RNA. So there's a specialized guide RNA that is required to target the site that you want to target because it matches the sequence of the locus. And it has specific properties so that it can identify the locus that you're trying to target. And it also brings the enzyme that's going to put in a cut in the DNA so the DNA can be repaired. I want to add that I'm using this general term guide RNA here or gRNA here, but with new technologies, there are new names for these RNAs. But essentially the function is the same, it does guide the enzyme to the site of mutation. So the design of this guide RNA or the gRNA is really important because that's what allows precise targeting of the DNA site that we want to edit. All the edits that are done, like I talked about, general gene editing, base editing, prime editing, all of these require an enzyme because what it does is it cuts the DNA. And that cut is important, or like it changes the base, and that function is important. So enzymes are needed, and it may require a correction template. So the question that was asked before, in terms of a deletion or an insertion, a correction template would be required. So there would be need for a guide RNA, there would be need for an enzyme, there would be need for a correction template. So you can see, like, if you need to add 3 different types of molecules, they have to be packaged together so they can get into the cell. The more types of molecules we add, the more complicated it becomes. But again, theoretically, it's possible. And so what happens is that the guide RNA allows targeting of the genome editor to the site of mutation. And this enzyme will either change a base if it's a base editor, or it can cut and lead to repair of the other enzymes come in to repair the DNA. And the mutation is gone and the actual correct sequence is present now. And once it's present, then a fully functional protein should be formed, just like it would in a normal cell. There are challenges and considerations when people are thinking for DNA editing. And those are that because there's so many different types of mutations, like hundreds of mutations in the CHM gene, different guide RNAs are needed for each of these different mutations. And their testing is important. So it's important that it's tested for its specificity. It should not bind to other places in the genome and lead to cutting at or edits at places where it's not supposed to edit or make any changes. For some of the mutations, designing the guide RNA can be challenging because of the adjacent DNA sequence. And again, depending on how the nucleotides are set, a proper guide RNA might be like really difficult to design. Another important consideration for DNA editing is that it should be active for like a short period of time. So it should be transient. It should do its job and it should go away, right? Because we do not want the enzyme and the guide RNA to hang around when the DNA has already been corrected, because there is a potential it can bind to other places in the genome. And so we just wanted to act for a short period of time, act efficiently so the mutation is corrected, and then for it to like go away. And so that's why a lot of people are focusing on, instead of providing gene for the enzyme and the guide RNA, they are focusing on providing the RNA itself and the protein itself so that they can get degraded in the cell and they don't like hang on, like they're not around for off-target edits. And it's also important to rule out off-target edits. So a lot of testing needs to be done beforehand to make sure that it's not going to cut the DNA at like, other sites, which is not intended. So that's about DNA editing.
Dr. Sinha, we have a couple more questions before
Sure.
we continue. Is that okay?
Absolutely.
All right. So Dr. J.R. Rizzo, who sits on our board, he actually has a question. If these can be used for only early and mid-stage choroideremia, how do you define mid-stage, and when would they no longer work?
So the mid-stage part is a little hard to define, but you can imagine that there should be enough cells available so that if you restore their function, partial vision can be restored, right? Or at least the individual will not continue losing vision. So as long as there's enough cells available, and the person has good enough vision, that's good. There are multiple tests that can be done that can, give an idea of like, oh, there's a significant loss of cells, and this therapy is not going to work anymore. So it comes down to preservation of therapy, preservation of vision, restoration of like, significant vision would be difficult, but if the cells are there, maybe it's possible. So mid-stage, again, it comes down to, like how the individual is seeing, and then how many cells are left.
We have another question from New York, from Orally. She's asking, what does it imply to design guide RNA?
That's a very good question. I didn't go into the technicalities of it. However, when a nucleic acid is made, the length of the nucleic acid matters. What's the target site and how many nucleotides are on each side, those things matter. What's the composition? For example, is there more of G and C type of nucleotides, or is there more of A and T nucleotides? Those things affect the specificity of the guide RNA. And so essentially designing a guide RNA means that you look at the sequence of the DNA that you want to edit. And based off of that, you see like, okay, I have to focus on this particular site. How much to the left or to the right, I need to include more nucleotides. So it's going to be specific and it's going to be efficient. And there are softwares available that can be used. So a DNA sequence is entered, and then it'll give out a few different types of guide RNAs, and it usually ranks them. And then it comes down to making sure to test them so that they are effective and efficient.
You can proceed. We're good. Thank you.
Sure. So moving on to translational read-through. Again, the topics that I'll be talking about, what is read-through? How does it target nonsense mutations? And can all nonsense mutations be overcome using the read-through approach? So again, based off of a previous question, we're just talking about nonsense mutation, where a meaningful codon has been changed to a stop codon, and the cellular machinery doesn't have a tRNA for that. And so protein synthesis stops there. And so can all types of nonsense mutations be overcome using this approach? And what happens in translational read-through? It allows continuation of protein synthesis instead of a premature termination. It can be done using two different approaches. One is the use of pharmacological drugs, and second is the use of engineered tRNAs. So the transfer RNAs that I talked about before, that bring in these building blocks for protein. And so with the pharmacological drugs, what happens is that when there's a stop codon in the messenger RNA and the translation stops prematurely, and we added a read-through drug, what it does is it can allow binding of tRNAs. It's not very random, but it is similar to the stop codon. There is no tRNA for the stop codon, but for example, if UAG is a stop codon, it might bring in a tRNA that has UGG instead of UAG. And so it's bringing something similar. So whatever amino acid that transfer RNA brings, because this drug is allowing not very specific binding of tRNA. So an amino acid would be inserted. It will not be the precise amino acid that's supposed to be there, but it will get inserted and the protein synthesis will continue. The hope is that this amino acid that's inserted is either the correct one that's supposed to be there or something that's not going to majorly affect the protein function. So there are read-through drugs that can prefer one type of amino acid over the other. So depending on the mutation, those could be tried. But this is how it works. It does not provide the exact amino acid, but it provides something similar. And the other kind of approach is the use of engineered tRNAs. It's a brilliant approach that was designed in Dr. Chris Ahern's lab at University of Iowa. And what it does is that I told you there are no transfer RNAs in a cell that can read the stop codons. But what if you engineered the tRNAs that can read these stop codons and put these tRNAs in the cell? So what happens now is that this engineered tRNA will bring the precise amino acid it's supposed to bring, and then a fully functional protein is formed. The only thing to keep in mind is that depending on the nonsense mutation and the type of amino acid that needs to be put there, the engineered tRNA will have to be slightly different. But there are not a lot of nonsense, like there are not a lot of different types of nonsense mutations. So there'll be only a few types of engineered tRNAs that need to be designed. And each individual will get like their specific kind of engineered tRNA. And so a full-length functional protein is expected to be formed. And again, just like with DNA editing, so that's just the basic overview of how the process works. Just like DNA editing, there are challenges and considerations that need to be had for the translational read-through approach as well. As you can imagine, as with any drug, there may be untoward effects. The other consideration or point, a challenge may be that read-through may not be efficient, for nonsense mutations that are very close to the native stop site. So where the protein synthesis is actually supposed to stop. If there's a nonsense mutation close to that, that might prove challenging because of how the cellular machinery recognizes the native stop. And so those kind of mutations are expected to be very difficult to treat using these therapies. And then because the changes are not happening at the level of DNA, so RNA is continually made in cells and it's degraded, new RNA keeps on being made, same for the protein, right? And because the new RNA continues to be made, the expression of these read-through drugs or the presence of the read-through drugs or expression of the engineered tRNA for long-term would be required. It cannot be like a one and done kind of a thing, or as long as there's a way to slowly keep on the function going, that'll work. And so in conclusion, choroideremia is caused by mutations in the CHM gene that encodes Rab-Escort protein 1 or Rep1. There are mutations, multiple different kinds of mutations that can lead to dysfunction of Rep1 or absence of Rep1. And today I talked about two different kinds of potential therapeutics, like DNA editing that can edit at the gene level, and translational read-through that functions at the level of the RNA. And these have the potential to be used as therapeutics for choroideremia. Just like with any therapeutic, it's really important to test delivery and efficacy to make sure that the molecules that we're using are functioning as we expect them to function. And there are preclinical testing platforms that are very important. So before anything goes to clinical trial, there's a lot of steps that need to be performed. So a lot of researchers are doing in vitro testing. We do a lot of testing in stem cell derived retinal cells. And then, eventually some, depending on the type of mutation, test in animal models before it goes for clinical trials. And so there's work to be done, but it's also promising that there are different avenues that could be tested for developing treatments for choroideremia. So in the end, I would like to thank my mentor, Dr. David Gamm. And I also want to mention I work, so Kim Edwards is a graduate student in the lab, and she works on choroideremia, and I kind of help her a little bit sometimes. And so, yeah, there are a lot of people in the Gamm lab who are working towards developing therapies for choroideremia and other retinal degenerative diseases. And then I want to thank Choroideremia Research Foundation for the funding we have had. And also last year we got the $1 million bike ride grant to study the engineered tRNA in collaboration with Dr. Chris Ahern. So I'm thankful for these fundings. And I do want to mention that, Choroideremia Research Foundation is doing such an important job bringing all the information to the CHMers and the families, and also keeping this connection between researchers and people who have choroideremia, because it helps us to, like, it pushes us to, and we know like how important all of this research is, but they're also a really good resource. So I'm glad that they started this webinar series. I'll be joining the other webinars too to learn more about potential therapeutics. So thank you all for being here today, and I'm happy to take more questions.
And we have them. So we have some coming in. So here's the first one. How far away is genetic editing in humans? Anne is asking.
So there are some clinical trials ongoing. It's for different diseases. And again, anytime it comes to modifying the human genome, it's we want to make sure that it's properly tested and making sure it's as safe as possible. So there are some clinical trials ongoing that can be, the information is available. It may take a few more years, but it's very exciting, all the new technology that's coming and how fast the field has moved forward in terms of making it more precise, making it more efficient. So it's hard for me to predict. I cannot predict exactly when, but the field is moving at a very fast pace. So I wouldn't be surprised if in next 5 years or so, there's more clinical trials, or looks like it has advanced a lot more than we expected.
Yeah,
but
there
it's to be done.
Good. That's good. Lisa asks again a couple more questions. What are the top 3 or 4 mutations that scientists are focusing on currently for therapeutic therapy? The most common mutations being looked at for therapeutic therapy? And then how far away are we from being able to apply these therapeutics to actual patients, which you just talked on?
Yeah. And again, usually if you can think of, if it's a simple mutation, like if it's a single nucleotide change, it might be simpler to target as well. People are looking into, like, for example, we are also looking into this nonsense, the read-through therapies for nonsense mutations, again, because almost 1/3 of patients are affected by that, right? But delivery, to make sure these molecules are delivered, is also an important question. So that's an important part. It's hard to exactly say what mutations, but I want to say people are exploring nonsense mutations, trying to target that. And if it's like single base changes, those can be comparatively simpler to treat. So those two, as far as I know, people are focusing more on. Insertions and deletions can be a little bit challenging, but again, like I said, the field is moving fastly, and if we can figure out a way to package this correction template, it would be possible to correct the gene as well, even if they're like big, like multiple nucleotide changes. And what was the other question? Like where the therapies, again, all I can say is that, researchers are working hard. We're trying to move things forward as fast as we can. There are some limitations with what techniques are available now, where the technology is. And there are a lot of challenges I did not talk about today in terms of like how we deliver, what kind of vehicle do we use, like how much do we deliver, what kind of models we use. But I want to let you know people are working on those questions.
Nama's joining us from Israel. So we have a couple questions from her. How can you tell if the mutation is close to a native stop site? And then here's another one. Is it in the number of the mutation sequence, for example, C887?
Yeah, yes. So for example, say, and again, there's no like a cut and dry like answer that exactly 5 amino acids before is the exact cutoff. Again, it depends on the sequence and where the termination complex binds when the protein synthesis is supposed to stop. So I'm just giving an example. It's a made up example. It's not an exact example, but assume that there are 1,000 amino acids in the protein, meaning there are 3,000 bases because it's 3 times in the nucleic acid, 3,000 bases. So if it's in the middle, say if it's a 1,500 base change, most likely it should be treatable. But if it's say at if it's a 2,950, it's so close to the end, it might be difficult to target that mutation.
She has a follow-up question saying, how can you produce long-term expression in the eye of the ACE tRNA? Thank you for an amazing talk.
Yeah, thank you. So one of the approaches is you can think of it just as gene therapy. So as you are providing using viral vectors, the idea would be to use viral vectors. But we know that based on some studies, it's not that it's going to be forever, but it's the longest of using other kind of approaches. So the answer would be any kind of DNA. If it's if the engineered tRNA is provided to a cell in form of a DNA, like it could be gene in a virus, or it could be like a plasmid if it can stay stable in the cell once it's delivered. So as long as it's in the form of a DNA, it should be longer term as compared to any other form. Like if we were to directly provide the engineered tRNA, that will degrade much sooner than the DNA that encodes the transfer RNA.
More questions. Can read-through be used for deletions?
No, it cannot be used for deletions. That's a general answer. And I say that because, as I mentioned, say you're reading a sentence and the words are in groups of 3 and it makes sense. But if you were to take out like 1 or 2 letters or 2 or 3 words, now even though you can keep going, it may not still make sense. In some deletions, what happens is that there's something called frame shift. So say the cell is reading in groups of 3, and as it keeps going, there was a nucleotide that was removed, that was deleted. So the RNA doesn't have that nucleotide. So the frame in which it's reading, it shifts. And in that case, a lot of times there's a nonsense mutation that's introduced a bit later on. So if the change in amino acid is not significant, then the read-through would work. But most likely the changes are going to be significant. Again, it's a case-by-case basis answer. So it truly depends on what deletion it is and what kind of changes are happening because of that deletion.
Yamil asks the question, would a single base replacement require a correction template? I have a CHM base mutation where C changed to T on exon 11. Would it require a correction template to be corrected?
It should not require a correction template. If there are base editors that can be used, it basically just requires the guide RNA to guide the enzyme, and the enzyme can change the base to what it's supposed to be. I do want to mention that for each of different kinds of bases, what it needs to change back to, there are different base editors, and some are more efficient than the others. Yeah, I just wanted to mention that, but yeah, a correction template technically should not be required if a base editor is being used.
Kayla is asking a question. When will this therapy become available? I have a 13-year-old who was diagnosed a few years back, and I haven't found any clinical trials for anyone younger than 18.
Yes, and yeah, that's a tough question to answer because again, we are making progress, but some of the things, again, we want to make sure the treatments, even if it's clinical trials, it's safe for the individuals that are getting it, right? And make sure that the chances that it works. So there are a lot of testing that goes behind the curtains, and once all those testings are done, then it moves forward to clinical trials. So that's a tough question to answer, but yeah, I'm hoping it'll be soon, in a few years. Yeah,
That's always hard for the moms of the group here.
yeah,
And the patients too. I shouldn't just say that, but it's just waiting, and that waiting time is hard, but keeping abreast of all this really good information. And especially with our organization and clinicaltrials.gov, it's a way to get on waiting lists and just be ready, be prepared, get in the queue. So that's good advice. I think this is the last one. Oops, maybe not. Let me just double check. Oh, Yamil says, thank you. It was very informative. We're going to close that one down. And then Orally says, thank you so much. Such a clear presentation. And first time I've ever heard of gRNA this way. In your lab, what is the influence or communication between your work and other labs working on metabolism and other types of research? And do you have any conversations on the relations between the gene, the cell effect, and the process?
yeah.
That's a lot. Do you want me to say it again?
Yeah, no, it's good. I think I understood the question. So that's a good question because, we do talk about all these different kinds of therapeutics and so focused on mutations. But we also know that individuals sometimes having the same mutation can have different disease progression. And what causes that? So it's not very clear. Could it be the lifestyle? Could it be some other factors in the genome? So it's really not very well known. I know people are working on some case studies and things like that. But we do, as a matter of fact, I do some metabolism research but that was for the healthy cells, just to understand how those functions in a dish. We collaborate a lot with other labs, and we try to bring in their technology, their perspective, and how we can apply to our model systems. So we do that. For now, our lab is focused on looking at these particular therapeutics to see how we can target the mutations or correct them. But I think there are some other labs trying to understand how metabolism, cell health, those kind of things affect the progression of the disease.
And some of those things that we are funding, the CRF is funding, some of those researchers and labs that are conducting that work. Well done. For the folks that were watching, both of us, before we started, we're both a little nervous. We were a little excited and nervous. So you did well. Thank you so much.
Yeah, thank you. And I do want to mention a couple of things, I don't want to hype the hope too much, but I do want to say that there's hope, like people are working on things.
Mhm.
And we also like hope things work as fast as they, but we have to keep safety, efficacy, those things in mind. And also to keep things a little bit like, in perspective, it won't be that people will like gain 100% of their vision back. But if they can gain something like, if they can um, get better at reading, like do a little bit more of what activities they lost, they can't do anymore, that'll be a success. Or if they stop, like if they don't lose vision anymore, what they have can be preserved, that's a success too. So I just keep it in perspective both ways. So I'm hopeful. Yeah.
Great way to end. That just continues our conversation into next week. We just have all these comments saying, Kathy, our executive director, thank you, Divya did an excellent job of making extremely complex subject understandable. I would cheer that one on to patients and families. And, I think with the gene therapy that we have been through, it's just good to continue to encourage our members that a lot of work is happening right now. And Dr. Brian Balios will be joining us next week, a week from today, talking about gene therapy, where we've been, where we're going. And so we hope everybody can come next week as well. And with that, I think we're done. Thank you so much, Divya. We really are encouraged. Tell Dr. Gamm hello from all of us.
Absolutely. Yeah. And thank you all for having me here. It was a pleasure. Thanks.
Thanks, everyone. We'll see you next week. Bye.